Good morning, everyone. I'm Ningyuan Zhu, a field bioinformatics scientist from MGI STOmics APAC. In today's presentation, I will give an introduction of the latest Stereo-seq product updates and applications on cancer research. So I will give a brief introduction of principles of Stereo-seq and the latest product launch and data performance updates, and also some publications of Stereo-seq on cancer research. So Stereo-seq is a cutting-edge spatial multi-omics technology for biological and translational research. Currently, Stereo-seq remains as the highest resolution and largest field-of-view spatial single-cell transcriptomic platform on the market. And now we have over 110 established Stereo-seq labs and also more than 200 research articles published using Stereo-seq since 2022. The high resolution and large field of view features are powered by DNB-seq technology. Here shows the DNB-seq system. The Stereo-seq chips are produced from a large chip and are then cut into different sizes for various applications. And the high-density DNA nanoball pattern array with coordinate information enables the high-resolution features of Stereo-seq. Here shows a video of the Stereo-seq workflow. Here's the DNB pattern array, and we firstly prepare sections from tissue and then put sections on the chip, and RNA will be captured. And after capturing the RNAs, they will be sequenced, and then we will get spatial transcriptomic data for further explorations. So we use DNB-seq features as the foundation for spatially resolved transcriptomic technology with high resolution and large FOV as follows. A standard DNB chip have spots with approximately 220 nanometer diameter and a center-to-center distance of 500 nanometer, providing up to 400 spots for tissue RNA capture per 100 square micrometers. DNB templates containing random barcodes and deposit on the pattern array, incubated with primers and sequenced to obtain the coordinate identity CID, which contains the spatial information of every DNB. And next, unique molecular ID, also called MID, and the probes are prepared for RNA capture. STOmics product offering range from mRNA to total RNA analysis, as well as RNA combined with the low-plex and high-plex proteins. We have two major solutions for fresh frozen and FFPE sample, respectively, and we provide solutions for different tissue sizes from small capture areas for organoids and biopsies to large capturing chips intended for profiling whole organs or embryos. We address multi-omics and multimodalities by ensuring compatibility with H&E staining for tissue morphology studies and also the MIF for the identification of specific protein markers, which are co-analyzed alongside high-resolution RNA profiles. And the transcriptomic solutions are species agnostic, which support multiple kinds of tissue types. Here's the example shows the chip sizes of our products. The standard solution includes two chip sizes, which is a 1 by 1 centimeter chip and also a mini 0.5 by 0.5 centimeter chip, which offers a more cost-effective option for smaller samples. And in addition, we offer a large chip design, which is with a standard maximum area of 2 by 3 centimeter. And also we have a customized chip, and the maximum area could be 13 by 13 centimeter, which is applicable for different tissue sizes. And currently we support large chip design for FF samples, and the FFPE large chip design will be launched soon. And STOmics provides end-to-end solutions for spatial transcriptomics, from biochemical workflow profiling to sequencing and bioinformatics analysis. In this workflow, tissue sections are loaded onto the chip surface, followed by fixation, permeabilization to capture the tissue RNA, and finally reverse transcription plus amplification. And once the cDNA is collected, used as template for library preparation, and sequenced together with the CID. And then the computational analysis of the sequencing data allows high-resolution, spatially resolved transcriptomics. We have developed two software packages called SAW and StereoMap to facilitate downstream data interpretation and analysis of sequencing outputs. And in this year, we have some product updates which improve the workflow and data performance. Here's the product roadmap of Stereo-seq in 2026. And in January, we have launched the Stereo-seq OMNI V1.1 for FFPE samples. And in Q2, we plan to launch the Go Spatial for Stereo-seq, which is the transcriptomics experiment automation system. And in Q3, we plan to launch the large chip design for FFPE V1.1, filling the gap of more chip sizes. And in the updated design of Stereo-seq OMNI V1.1 for FFPE samples, it utilized the combinatorial probes that can detect both coding gene and non-coding gene, and also the microorganism genes. The Stereo-seq can achieve single-cell resolution. Since the average diameter of a cell is about 10 micrometer and the distance between two spots is 0.5 micrometer, so each cell can contain about 400 spots. So researchers could use different bin sizes based on tissue type, the data quality, and their own needs. We use bin size defines the spatial resolution by grouping spots into larger pseudo-spots, a square unit. For example, based on the 10 micrometer diameter of a typical cell, we standardly use bin 20 to merge spots together, and each spot have a length of 10 micrometer, correspond to the cellular scale. And furthermore, to enhance the accuracy, we utilize the custom cell segmentation algorithm that refines this process, enabling precise per-cell data summarization. So despite the single-cell resolution, the Stereo-seq OMNI 1.1 solution for FFPE samples are also compatible with low-quality samples, which DV200 is more than 30%. And also, this workflow don't need for tissue permeabilization. With this latest version, V1.1, we have focused on 4 key areas. The first one is 1 to 3 times higher capture efficiency to provide you deeper insights per experiment run, and also the preserved spatial fidelity with reduced tissue diffusion to ensure true single-cell resolution. Also, we have improved the data usability to efficiently harness information within clinical samples and the extended capability to sequence together with fresh frozen Stereo-seq libraries to enable a more user-friendly application. So together, these innovations mean that every run generates richer, more actionable biological insights. Here shows an example of data performance for the latest version of OMNI FFPE. We can see the bin 20 median gene is about 420 in the latest version compared to 155 in OMNI FFPE V1.0 using the same sample. And the median gene count in bin 20 level is 73% higher than the old version. And the improved single-cell analysis enables well-defined tissue annotation and stratification. We can see the clear spatial distribution of each cell types in this mouse intestine data. And we also provide an optional workflow to remove ribosomal RNA. Adding a commercial rRNA duplication kit reduce rRNA to nearly 1%, increasing transcriptomic percentage by over 15%. So our solution is designed to work across a broad range of tissues, from brain and kidney to skin, thymus, and even some challenging clinical samples such as lymphoma, liver cancer, and TMA samples. With the improved capture efficiency and the reduced level of diffusion, clinical sample multiplexing on the same chip has now become possible with Stereo-seq OMNI 1.1. So you are not limited to one disease area or one tissue type. OMNI 1.1 can support and accelerate your discoveries. We can apply both TMA FFPE block and multiplex samples that mounting multiplex sections on one slide. Here's an example of data performance of these samples by our R&D team. And we can see the data quality is able to be performed for downstream analysis. And also, because FFPE solution is able to detect microorganism genes. And here's an example of the host microbiome co-detection in mouse intestine samples. In previous study, we have reported 73 common gut microbiome species in normal mice. And in our data, we have detected 23 species by OMNI 1.1 in the small intestine samples, accounting for about 62%. So FFPE V1.1 solution empowers breakthrough across various research domains. In cancer study, we're able to identify some biomarker using the Stereo-seq data for predict some markers for subcell types. And also we can conduct the whole cohort scale spatial analysis for large data set analysis to see the prognosis and some clinical outcomes. And also because some non-coding RNAs play an important role in cancer development, we can focus on link RNA modulation for cancer study. Here's an example use tumor samples for Stereo-seq data analysis at single cell resolution level. We can see some major cell types are annotated in this sample, and we can see the heat map of marker genes shows a clear pattern in each cell type. And the clustering and CMV analysis shows that it is consistent with the H&E staining image. And also the hallmark pathway enrichment and cell neighborhood enrichment analysis reveals the distinct spatial clustering patterns between tumor and non-tumor regions. So we also have other products of solutions for fresh frozen samples. For the latest version of FF Solutions V1.3, we offer higher capture efficiency and which enables single cell analysis across the unbiased whole transcriptome. Also, it supports largest capturing FOV for large tissue sizes. In this case study, we demonstrate the Stereo-seq's ability to reveal the pathological changes in human esophageal cancers. We can see a clear observation of tumor cells infiltrating the basal layers of the esophageal tissue. And the squamous epithelial cells were observed to be surrounded by fibroblasts, indicating the invasive characteristics. So Stereo-seq FFV 1.3 allow us to uncover and recognize new insights. Using this spatial single-cell analysis, we are able to precisely annotate the cells and gather spatial cellular data. We can see the spatial pattern of cell types are matched with the H&E staining image. So this method also uncover a rare cell type, glandular epithelium within the tumor area. And our examination of spatial molecular data demonstrate the cellular features that harmonized with the histological and diagnostic attributes. So despite these two major solutions, we also have another multi-omics approach involves incorporating multiplex IF assay into the Stereo-seq transcriptomic workflow for improved identification of some biological protein markers. This is suitable for low-plex protein and transcriptomic code detection. So in this solution, we support RNA transcriptomic detection and 4 IF channels for protein detection. In this sample of the mouse brain sample, the tissue was stained with DAPI, neuron, and SMI32. And can view more examples, download the demo data from our official website. And another solution called Stereo-seq proteotranscriptomic solution is designed for simultaneous protein and RNA code detection. And with this solution, researchers can detect over 100 protein targets alongside whole transcriptomic profiling. And this is powerful for studying immune cell interactions and cellular environments. And this integrated approach allows for a more comprehensive understanding of both gene expression and protein level signaling within the same tissue section. So the Stereo-seq solution uses commercially available antibodies sourced from BioLegend. You can choose between the premixed antibody cocktail panels for broad profiling study and also the individual antibody panels for more specific focus studies. So currently, the Stereo-seq solution is only validated for human and mouse samples. And for details on supported antibodies, please refer to the resources available on our website. And this example demonstrates the powerful co-detection capability of the Stereo-seq proteotranscriptomic solution. So we can see the whole transcriptomic data together with over 100 proteins and without compromising mRNA capture efficiency. So here shows the spatial distribution of some proteins, and we can see we can integrate the RNA and protein data together for downstream analysis. So that is the introduction of the latest product updates of Stereo-seq. In the next session, let me introduce some applications of Stereo-seq on cancer research to see what kind of analysis Stereo-seq can do in cancer study. So the previous as we mentioned before, the Stereo-seq is able to detect the microorganisms, and this research is conducted by our collaborator, Professor Patrick Tang from Singapore Duke and US Medical School. And in this case, they utilize Stereo-seq for bacteria capture. This research focused on intestinal metaplasia in gastric cancer, and they performed whole genome and methylation sequencing analysis, identifying mutational signatures related to gastric cancer. So for the microbiome detection, they used fish experiments confirming the presence of a kind of bacteria called Streptococcus in gastric cancer tissues. In Stereo-seq data, they detected spatial clusters of bacterial reads mapping to Streptococcus, and in one gastric cancer sample, these Streptococcus regions overlapped with areas exhibiting elevated expression of CXCL8, a key mediator of inflammation and suggesting a local activation of inflammatory pathways. And in the other sample, they also found the Streptococcus overlapped with IgA plasma cell signatures, consistent with the increased inflammation and cancer progression. And in Professor Patrick Tang's another paper also published on cancer discovery, they used GeoMx DSP and single-cell RNA-seq to analyze the gastric cancer samples and classify data into 2 subgroups, G1 and G2, and shows different invasiveness status. So to enhance the spatial resolution and ensure the reproducibility, they applied Stereo-seq to 2 GC samples, successfully identifying the G1 and G2 subgroups with similar pathway enrichment as seen with the Geomics DSP, and which demonstrate a high resolution of Stereo-seq. And here also shows an example of another application. This research is conducted by BGI team and our collaborators, and they use Stereo-seq to comprehensively profile TLS characteristics and their interplays with the tumor microenvironment. So TLS, the territory lymphoid structures, are ectopic lymphoid aggregates found in numerous cancers, and often linked to enhanced immunotherapy responses and better clinical outcomes. The mature TLS harbor germinal centers essential for B cell development and function, and it serves as the predictive biomarkers for immunotherapy. So in this case, scientists categorized a mature TLS into conforming and deviating TLS, which demonstrate different immune response to anti-PD-1 treatment. This research investigated the metabolic pathways affecting TLS development and proposed potential strategies for tumor treatment. This research includes 46 fresh frozen samples from both treatment-naive HCC liver cancer patients and anti-PD-1 treatment patients. They were sequenced using Stereo-seq, single-cell RNA-seq, and SN-ATAC-seq to perform a multi-omics analysis. To identify the TLS regions, they developed a computational strategy based on spatial transcriptional properties and validated with gene module analysis, H&E staining image, and also the MIHC images. So to dissect the primary cellular composition, they performed the spotlight at bin 50 resolution with a spot length of 25 micrometers. And this cell type annotation is aligned accurately with the anatomical structures, reveals that TLS are highly heterogeneous cells and vary in cellular composition and spatial organization. So to elucidate TLS developmental states, this research developed a spatial transcriptomic-based strategy to classify TLS as conforming or deviating from the maturation process, constructed a benchmark differentiation trajectory from naive B cells to germinal center B cells, describe their unique characteristics in terms of gene expression, cell composition, and spatial patterns. So they identify marker genes for the development process, and these results also confirmed by MIHC images, elucidate the molecular mechanisms underlying TLS maturation, and provide a valuable resource for characterizing TLS lacking key maturation features. And then with the mature TLS defined by high CR2 expression, they used linear regression analysis and determined whether TLS conformed to or deviated from the established maturation trajectory. They also developed a convolutional neural network model to categorize the immature TLS into conforming or deviating groups. And to further validate the TLS classification, they utilize cell-align, which aligns the developmental trajectories to compare the expression dynamics between developmental trajectories of naive B to GCB cells within mature, conforming, and deviating TLS categories. So this result reveals the distinct spatial properties of TLS with various developmental states. And they then assess the prognostic value of TLS subtypes in HCC, revealed that deviating TLS may be related to higher risk scores and worse clinical outcomes. and the pathway enrichment analysis and as single-cell RNA-seq data validation revealed that conforming TLS but not the deviating TLS exhibit a robust response to ICB therapy. And to further investigate the molecular mechanism of TLS subtypes, scientists performed trajectory-based differential expression analysis to see the dynamic transcriptional differences in naive and GCB cells across TLS subtypes, identifying more than 1,000 differentially expressed genes between mature and deviating TLS. This result suggests that altered metabolism may play an important role in the misregulation of TLS maturation. And then they compared metabolic activity in naive B and GCB cells with deviating versus mature TLS, suggests that the aberrant tryptophan metabolism may disrupt their differentiation trajectories. So to evaluate the driver of the metabolic heterogeneity, they use PBMC single-cell RNA-seq for validation, indicate that the tryptophan metabolic heterogeneity is acquired from the tumor microenvironment rather than the intrinsic developmental process. And from single-cell data, they found malignant cells contribute to the aberrant metabolic milieu. And this finding are also validated by single-cell ATAC-seq and also validated across multiple cancer types, suggest that the elevated tryptophan metabolism in TLS may be a general factor affecting TLS development in tumors. And in the end of this research, they used an immunogenetic HCC mouse model for experimental validation, suggests that the tryptophan or its metabolic products play a suppressive role in TLS development in HCC. And the restricting tryptophan metabolism in those synergistic effect with anti-PD-1 treatment in enhancing TLS maturation and anti-tumor activity. And another kind of application of Stereo-seq is to focus on different cell subtypes. In the last paper, we can see they can detect subtle kind of spatial structures. And in this research, this is another research article published on Nature Communications by BGI team and our collaborators. And this research applied Stereo-seq to build a comprehensive high-resolution spatial map and characterize the geospatial architecture and the cell-cell communication in 30 primary breast tumors and paired lymph node metastasis samples in order to optimize the immunotherapy efficacy. So in this study, they provide a detailed spatial landscape of human breast cancer and offering key insight into the granular regulation of tumor immune infiltration. So at the beginning of this study, scientists utilized the RCTD algorithm to infer the cell composition, also at bin 50 resolution, with internal and external single-cell RNA-seq data sets as independent reference. And this reveals a detailed distribution of various cells in primary tumors and metastatic lymph nodes, enabling further characterization of the subtle structure of the TME. So to further investigate the immunomodulatory function of granular endocellular cells, they classified the PKM1 positive ECs into 3 categories, characterized by distinct endothelium identity markers using 2 independent clustering methods. And the differentially expressed gene analysis suggests a common immunomodulatory role for granules in the breast tumor environment. And to describe the microenvironment of different endocellular subtypes, They determine the cell composition in each bin50 spot containing endocellular cells. And also they quantify the proximity relationship between endocellular cells and other major cell types by the enrichment scores to see which kind of cell type is near the endocellular cells. And combining with in situ projections on the Stereo-seq slide, they found the capillary ECs and tumor cells are colocalized, and the venular ECs are located in those proximity to T and NK cells. So these distinct structures were also identified as a TLS-like organization. And to illustrate the synergistic interaction between venular endocellular cells and the smooth muscle cells in the regulation of immune infiltration, scientists characterized the heterogeneity of smooth muscle cells. With the validation using single-cell RNA-seq and the MIF staining, they revealed a spatial co-localization between CCL21 positive SMC and also ACKR1 positive ECs. And in this spatial map, they observed a characteristic annular distribution of the arterial SMCs along the outer perimeter of the arterial ECs, highlighting the precise localization achieved through Stereo-seq. And to explore the clinical significance of EC and SMC subtyping, the research team performed a single-cell data analysis in 2 large breast cancer cohorts, and they find subtype-specific signature genes. This analysis indicated that a high proportion of venular ECs or SMC was significantly correlated with improved overall survival. And this result took other factors into the calculation, such as age, tumor stage, and ERPR, HER2 receptor status. And what's more, they performed the gene module identification and the cell-cell communication analysis reveals that the ACKR1 ECs and the CCL21 SMCs coordinate to promote immune infiltration. And so at the end of this research, they built a hypothesized model for the collaborative function between venular SMC and the endocellular cells to augment the lymphocyte extravasation. This study explored the potential therapies that can induce the formation of venules within the tumor. So in a summary, the advantage of Stereo-seq is that it is able to detect some subtle structure like TLS, microorganisms, to describe the distinct spatial characteristics and tumor microenvironment. And also Stereo-seq can help us identify some specific cell subtypes. And with the integrated multi-omics analysis using other technologies such as single-cell RNA-seq and histological data such as H&E and MIF staining image, we can get a broader insight to further investigate the molecular mechanism of cancer development. Also can explore the therapies and the relationship between specific cells and prognosis. Okay, so that's all of my introduction of the Stereo-seq products and its applications on cancer study. Here's our STOmics website, and please contact me, email me if you have any questions about Stereo-seq. Thank you for listening.